I'm going to talk about AI in pharmaceutical industry. And you know AI is a relatively nascent field, and human biology is extremely complex. So the use of AI in human biology and drug discovery, there is a lot of speculation, but time will tell what would be the reality. I'm coming from Europe, and I thought I'll start with a story of Madame Jean Calment, who was a very spirited lady who lived in the center of Paris. Madame Jean Calment did a deal with her lawyer when she was 90 years old. And that deal was unusual for that time. Reminiscent of the reverse mortgages that we know, the lawyer agreed to pay Madame a fixed amount every month till her death. In return, the lawyer could keep her very nice apartment in the center of Paris. So that was a deal. The lawyer, being a lawyer, did his due diligence. And he thought it's either a good deal or a very good deal. Because Jean Calment was 90 years old, well above the average life expectancy in Paris or France. And she was a chronic smoker, even at that age. To cut the long story short, she outlived the lawyer and the lawyer's son, and the lawyer's family ended up paying her more than twice the value of the apartment. She continued smoking till 117 years old, and she stopped not because of health issues, because her eyesight was getting diminished and she couldn't light the cigarettes, and she needed help, so she just gave it up. At her 120th birthday, when there was a small party, a few journalists came to her, and a young French journalist came to her and said, "Madam, we hope to see you next year too." And she looked up and said, "Why not? You seem to be in perfectly good health." She lived for another 2 years, and at the age of 122 years and 164 days, she still holds the world record for living the longest. Now, we know that this is an exception, not a rule. But does it have to be an exception, and can it be the rule? Jürgen, when we were talking during the breakfast, he was talking about a longevity expert who said that we can live till 120 years. And this is where that number roughly comes from. Now, if you look at the purpose of the pharmaceutical industry, it's obviously to prolong lives, but it's also to make sure that we age healthy. As you know, when we age beyond a certain point, the quality of life drops down, the disability associated with age goes up, and therefore the overall health quality in the later years are not optimal. So the goal is, of course, to help live longer, but also healthier, something I am sure everybody would agree. Now, I know this audience is more tech-centric, so I try to use an analogy to describe how human biology works. And let's take this construction site as an example. There will be a master blueprint for the construction, and there will be people, supervisors who makes copies of these blueprint, goes around to different parts of the construction site and help give instructions. And then there will be workers who really does the work, yeah? So 3 parts. So human biology works something like this. We have a master blueprint called as our genes or the DNA. Then, like the supervisors with the copy, we have something called as a RNA. RNA is copied from the gene on a regular basis. Sometimes it's turned on, sometimes it's turned off, and there are various levels of expression, but that's the way the body regulates it. And then the construction worker who really do the building are proteins. Yeah, so the DNA, RNA, proteins, that's the way human biology works. Now, if you look at all the drugs and the medicines in the world, more than 90%, probably more than 95% of all of them targets proteins. You look at Lipitor, it targets an enzyme, HMG-CoA synthase, it's a protein. You look at aspirin, it targets cyclooxygenase, it's a protein. You take insulin, it itself is a protein, right? You take, Diovan, it works on angiotensin receptor blocker, it's a protein. So vast majority of the drugs target protein. We were not able to target the genes or the RNA in the past, but the recent advances are changing it. So the druggable universe is significantly expanding than in the past. Even the proteins, not all the proteins are druggable. So even the druggable universe is limited, but now we have RNA and the DNA. Let me give you some examples of that. So first, I'm going to talk about something called as gene therapy. And gene therapy is basically when something goes wrong with the blueprint. Can we address that? So I will talk about gene replacement therapy, where the blueprint is now working, the gene is not getting created, and we will give a gene into the body only once. Not like every day or every month, only once, and then the body continues to make that protein that is required. And last year, there was another new technology approved, which is called as gene editing. That means if there is something wrong with the sequence of the gene, you can just go and edit it and correct it, right? So that's another technology approved by FDA last year. Then I'll talk about cell therapy, where you take the cells from the body, you do genetic engineering and put it back, and that does some work. I'll give that examples. Let me go to these first two examples, and then I talk about the rest. Okay. So here, I'm going to talk about a disease condition called as spinal muscular atrophy. It's a rare genetic disease. The kids who get impacted by this disease are severely impacted. 60% of SMA patients have type 1 SMA, which is the most severe form. 90% of those patients die within 2 years, so very severe. So what happens? So let's say I decide to lift my hand. So my brain makes that decision. Brain communicates to my muscle through a neuron or nerve fibers, and then my muscle does the job. In patients with this condition, these neurons die very fast. When they're born, it's fine, but every day neurons start to die. And once the neurons die, neurons don't regenerate. So the kids who have this disease, they cannot breathe. Breathing requires muscle movements. They cannot walk. They cannot move their fingers sometimes. And they cannot even swallow, because swallow also requires muscle movements. And that's why a lot of these kids with severe forms die by the age of 2. Now, I'm going to show a video of Amelia, who has a less severe version called as a SMA type 2. That's why she survived. She's probably now 18 years old. And she is in the wheelchair. And you will see her sister who is running around. And I'll tell what's the difference between them after I show the video. So, Amelia has a very difficult life. Even though she's alive, she misses 50% of her schools. She has constant infections, and she has a lot of doctor visits, right? But look at her sister. She has exactly the same disease. She's running around like a normal kid. So what happened was, because Amelia had this disease, the doctor suspected that Lena, her sister, would also have that disease. So as soon as Lena was born, they immediately diagnosed for this disease. And they gave this gene therapy that I talked about for the SMN1 gene. And that is why she is like a perfectly healthy kid. So it's a question between life and death, almost a perfect life versus something like death or disability. And this is the latest picture from the Great Ormond Street Hospital in London. Lena is a little bit of a celebrity there. She's one of the early patients who got this gene therapy, and her sister, while disabled, still is a patient at the hospital. So these are the miracles that are happening in the field of biology and drug discovery. Now, for these drugs to be effective, you need to act early. Remember, time passes, neurons die. Neurons do not regenerate, so you cannot reverse the disease. So it's very important you diagnose early on and treat. And most of the time, it's too early, that it's too late to diagnose. Amelia was diagnosed after 1 year. She was not moving well, went to the doctor. Doctor said, it'll be fine, you're very worrying too much. So the diagnosis often takes time. Now, in many parts of the world, every kid that is born is screened for this disease immediately. So in the United States, almost all kids, unless you opt out, will be subject to a newborn screening. 20 plus countries in Europe has this. So Latvia has this as an example. And the moment a baby is born, they screen it, and if they suspect, even before any symptoms, they give the gene therapy, because you cannot or should not wait for the symptoms to appear, okay? Let me tell you another story. This is a little bit older. This is a patient called as Emily Whitehead. Emily was diagnosed with a condition called as acute lymphoid leukemia, a blood cancer. As you can see, she is getting chemotherapy from that machine. It's her father on the back. You can see, as a side effect of chemotherapy, she has lost her hair. She's very frail, and that's why she's using a walker. And after the chemotherapy used for 2 years, her disease relapsed. And the doctor said, you can go home and pray. There is nothing much more we can do. So the scientists in the hospital tried something radically new, which was never tried before. So they did this. They took the blood from Emily, and they took the T cells, which are the immune cells that attacks bacteria and viruses, and they re-engineered in the laboratory to teach how the T cells can selectively go and kill the cancer cells in Emily's body. So it's using Emily's immune defense and teaching that immune defense and say, okay, you know how to attack a bacteria. We are going to teach you how to attack the cancer cells that she has, because the cancer cells is a marker called a CD19 expressed in the surface. So they re-engineered that and re-injected to the patient. So what happened? They did this experiment, and Emily felt really ill. She underwent something called as a cytokine release syndrome. High fever, body was swelling, she has difficulty to breathe. And they put her in a coma, induced coma, and she was in ventilator for 14 days. And in clinical trials, when you have something bad like that, you start to write a grade 5 report. This is to tell the FDA, well, our first patient has died, and then the trial is immediately stopped, right? So the guy wrote the letter, but luckily he did not send it. The blood tests came back, and they saw there was a high level of a protein called as interleukin-6 in her body. And the doctors gave some medicines for interleukin-6, and then she recovered very well. And then, it's probably more than 10 years, she's fully cancer-free. it's complete remission. It's a cure of cancer in a case, and almost 80% of the patients in that trial had complete remission. So all the happenings after the therapy was a good thing. It was because the therapy was working. Because when the T cells went and attacked the cancer cells, it released a lot of cytokines as an immune response, and that's why she had this incident. But now we know how to manage it. Every time we do CAR-T, we give the anti-inflammatory drugs, etc., so it's become a standard protocol. Now, CAR-T is extremely beneficial in blood cancers. People have tried that in solid tumors. It has not worked well. And now there is a huge amount of interest in autoimmune disorders like lupus, like rheumatoid arthritis, etc. So there's a lot of work going on with CAR-Ts. But you know, it's a very complex thing. You need to take cells from the patient, send it to a lab, re-engineer, bring it back. So it's a very laborious process and also expensive because of that. So we talked about the other two. Then there is the RNA interference, siRNAs, antisense oligonucleotides. You probably also remember the COVID vaccine that came from Moderna, which was also at the level of RNA. Very briefly about the other two. You know, when patients have cancer, the standard treatment is radiation and chemotherapy. Yeah? Now, the antibody drug conjugates are trying to make it more targeted. Because when you get chemotherapy, I don't know if you know anybody who has gone through chemotherapy, it's a horrific experience. Yeah? You vomit a lot, you know, you lose your hair. It's pretty tough. So antibody drug conjugates, they have the chemotherapy, but it's linked to an antibody who can recognize cancer cells. So it only goes to those cancer cells and try to destroy the cancer cells. So the healthy cells are, by and large, less impacted. The other one, radioligand therapy, is the same thing. You have the radioisotope linked to a ligand that also knows how to identify the cancer cells, so the radiation is targeted to the cancer cells. So the first gene therapy and the second gene therapy is in the Novartis portfolio. The first cell therapy that I showed you is in the Novartis portfolio. RNA interference, we are not the pioneers, but we have multiple. Same with antibody drug conjugates. We have multiple drugs, but not the pioneers. A radioligand, we are the pioneers. The first radioligand therapy was approved last year for prostate cancer, and that is a Novartis product. So we are at the forefront of these innovations. Now, that's all great, but we have a problem. You all know the Moore's law, and where the computing power becomes faster and less expensive over time, right? So that's fantastic. Over time, things get cheaper and better. In the pharmaceutical industry, we have the Vroom's law, which is the opposite. As time goes by, it becomes slower and more expensive to develop drugs. So while we have all these fascinating new technologies, the productivity, the R&D productivity is suboptimal in our industry. And it is in this context that I want to talk about AI and what it could do to the drug discovery and development. So this is the stages on how we develop drugs. First, we discover a target. What target in the body do you want to modulate? Then we make a medicine. We engineer in the lab, a lot of chemistry. We try to attack the target. And then preclinical, first we do the work in animals. And if the drug is pretty safe and effective, we try in healthy volunteers, which is called as a phase 1 trial. Then if it is good enough, we try in the phase 2 and phase 3. Phase 2 is try to identify the dose mostly, and then phase 3 are the very large clinical trials in patients. And if the data is good, we go to the regulators like FDA in the US. And as you can see, the cumulative costs would continue to go, and it reaches roughly $2 billion to produce one drug. Why $2 billion? It's because a lot of drugs fail. So you also need to pay for the failures. Yeah? 1 out of every drug that goes to phase 1 gets approval. 9 fails. That's why we are thinking about how can we predict better which drugs will succeed and which will not. And AI will have a profound influence on all of these. And I will argue that the early impact of AI would be on the clinical trials, and the later impact will be in the discovery phase. But the later impacts, I think, will be much more profound. Let me explain what I mean. So if I look at what can happen with AI in pharmaceutical industry, there are things that can happen faster, the low-hanging fruits, but their impact will be relatively modest. And there are things that will take time, but their impact would be much bigger. So for example, in clinical trial operations, when we do this trial, AI can better predict which sites may have more patients, because we want to be fast in getting the drug developed. Or it can help the doctors identify which patients may be best responsive for this drug, so recruit these kind of patients very fast. So those kind of things can happen. Then AI can also, looking at the clinical trial data, think which are the patients who will benefit the most. Remember, we have drugs approved for a lot of diseases, but not every patient benefits the same. So identifying which subpopulations of the patients are more likely to benefit and target the drug for those patients is something that can happen also relatively straightforward. The other two are much more impactful. Can you use AI to design a drug? And more importantly, can you use machine learning to identify new targets, proteins, genes, because we still don't know how many diseases works. So this is a chart for the United States, 15 to 50-year-olds, and this calculates the years lost in disability. So these are conditions that cause ill health in this age group. And you can see a large portion of this is neuropsychiatric disorders. If you increase the age group, of course, heart failure and all those things start to come in, but you can see a large chunk of that is neuromuscular psychiatric disorders. Even you could argue that alcohol and drugs have also linkage to neurology and brain. And this is an area we don't understand the science. We still don't understand what causes depression. How does schizophrenia happen? Through serendipity, we have discovered some drugs in the past, and we kind of use it, but they are suboptimal drugs. And this is the area that will probably benefit the most from machine learning-enabled target discovery. Now, I'm going to focus the rest of the talk on the drug designs. Remember, I told you it's mostly proteins that are used for drug targeting. And first thing we used to do is to identify the structure of a protein. Protein has a 3-dimensional structure. It can be like this, like this, like this. Unless you don't know the structure, you cannot design a drug to go and block it, as an example. So you need to understand the structure of the protein. The way to do that was called X-ray crystallography. You make the proteins into crystals, you bombard with X-ray, you look at the patterns of which how this is scattered, and you form a map, and then based on that map, you speculate what will be the structure of the protein. Identifying structure of one protein was a PhD thesis. You spent 3.5 years doing just one protein. It's a Nobel Prize-winning techniques. And it was a very time-consuming. People do structures, put it in a database, then move to the next one. That's how the field used to work. And now, AI has learned to do it better and faster. The great example is Google DeepMind. They just need to know the sequence of the protein, which is very easy. They put it to the algorithms, and you get the structure of the protein. Very simple. Yeah? 3 years of work replaced probably by 30 minutes or 1 hour. This used to be the place that holds the conference, which was called as the Olympics of Protein Structure. This conference, called as a Critical Assessment of Structural Prediction, is a very nerdy conference, as you can imagine. Used to go and people tried to solve structures. For many years, the output was stagnant, and for the last 2 times, it started to leapfrog. Why? The AI was introduced. So you can see this was in the 2020 session. You could see the performance of the competitors. The number 1 was AlphaFold 2 team. Yeah? So the DeepMind team. Many of the people below also used AI, but they used different AI. The only person who didn't use AI came up 57th ranking. It is a very clear example of AI outcompeting human ingenuity, at least in this case. Now, DeepMind do not participate in this competition anymore, but everybody else uses DeepMind to do this prediction. So in the past, we designed drugs using something called as high throughput screening. Every pharmaceutical company has a big library of products. And once we know this is the target we want to hit, we will make sure we will try to get all these products one by one and see if it works or not. Now we are doing computer-aided drug designing. We are learning to iterate the molecules. But in the future, we are able to design drugs using generative chemistry. So in the past, we were limited by the compounds that we had in our proprietary library. In the future, AI, like companies like Isomorphic Labs, Recursion, they design molecules that doesn't even exist in the nature. They look at the protein and say, which is the molecule that can best bind and block it? But they also look at, would it create safety problems elsewhere? Would they be permeable into the cells? How would the pharmacokinetics? They can predict all of these things using generative chemistry. Designing molecules, and Mother Earth is a great chemist, and it has fantastic molecules already in store, but the future is about designing drugs unbiased by what is existing in the nature or in the libraries of pharmaceutical chemistry, and that is very, very impressive. And Novartis has collaborations with Isomorphic Labs as an example. Now, I want to remind you that when computer-aided drug design came, this was a cover of Fortune magazine in 1981. They said it's going to be the next industrial revolution. It helped, but it did not create a revolution. So we need to be mindful that, would it be a hope or a hype? We don't know. I have very good reasons to believe that AI is going to create significant impact, but time will only tell. These are the list of AI-designed molecules that are in clinical trials. None of them have reached phase 3. Maybe the first one is in phase 2, phase 3. The rest of them are in early stages of clinical trials. So we need to see what will go through and how many of them will make it. But there is movement there. So going back to the living longer and healthier, this is a chart showing the number of centenarians, people above 100 years, in the United States. So when I compare roughly to when I was born, so probably 10,000 centenarians, now it's almost 100,000. It's 10-fold increase of centenarians in my lifetime. It's a very big increase. You don't see that increase because we are projecting the future, and that curve is even higher. And this is even supposed to go much higher. So there is already a very strong trend of people growing a lot, living a lot longer. And this does not account into AI or any of the things that I have just said. It's just a secular trend and the projection of it based on some other factors. Madame Jean Calment, during her 100th birthday, somebody asked her, "Madam, what is the best thing about being a centenarian?" She said, "No peer pressure." But that will likely change thanks to the advances in medicine, but hopefully also enabled by the AI. I thank you for your time.